Hello everyone. Welcome to the fourth part of biological classification. In this video, we'll discuss kingdom fungi. Kingdom fungi includes organisms that are heterotrophs and have cell wall made up of chitin. They can be characterized as saprophytes, parasites, and symbionts. Saprophytes are heterotrophic organisms obtaining their nutrition from soluble organic matter of the dead decaying substances. Parasites depend on living plants and animals, whereas symbionts live in close association with any other species such as algae in lichens or with the roots of higher plants in mycorrhiza. To reproduce, but they may use vegetative means, asexual, or the sexual means. Vegetative reproduction could be either through fragmentation, fission, or budding. Asexual reproduction is through variety of spores, for example, conidiospores, sporangiospores, or zoospores. Sexual reproduction is through spores again, and they are special spores known as oospores, ascospores, or basidiospores. Members of kingdom fungi exhibit variety of ways to reproduce asexually. The first method is transverse fission, where formation of a transverse fissure lays down new cell wall between the two dividing cells. Second method is hyphal fragmentation, which is due to the fragmentation of hyphae in order to produce arthroconidea or arthrospores. Upon germination, they'll give rise to a new hyphae. Chlamydospores are of two types. If they are found at the apex of the hyphae, they are called terminal chlamydospores. They can also be found within a hyphae. Apart from these three, we also have sporangiospores produced in a sporangium. We have conidiospores born on the conidiophore, and we have blastospores that are produced by the vegetative mother cell. Sexual reproduction in fungi follows three generalized steps. The first step is plasmogamy, that is the fusion of protoplasm of two motile or non-motile gametes. Second is karyogamy, that follows plasmogamy, where the fusion of two nuclei takes place. And finally, it is meiosis in zygote in order to produce haploid spores. For plasmogamy, two haploid hyphae of compatible mating types are selected for fusion. In some fungi, the fusion plasmogamy may immediately be preceded by karyogamy to result into diploid cells. In many other fungi, like ascomycetes or basidiomycetes, there is an intervening dikaryotic stage that is represented by N plus N, or the two nuclei per cell. This means that the protoplasm has fused, but the nuclei is yet to fuse. This condition is called as dikaryon, and the phase is called as dikaryophase of fungus. Later on, when karyogamy occurs, the two nuclei fuse and the cells become diploid. The fungi forms fruiting bodies in which reduction division or meiosis occurs that leads to the formation of haploid spores. This is a schematic representation of how reproduction in fungi takes place. As you can see here, there is alternation of generation, haploid and diploid stage, and there is a special kind of stage, the dikaryotic stage in between in some fungi. So it all starts with the cytoplasmic fusion, the plasmogamy, wherein two gametes of opposite mating type, of compatible mating type, will fuse with each other, and this stage may or may not occur in different members of fungi. Wherever it occurs, there is fusion of protoplasm, but the nuclei is yet to fuse. So the condition is two nuclei per cell, N plus N. After the stage is over, there will be karyogamy or nuclear fusion, which leads to formation of zygotic stage. Zygote is formed, and reduction division, this enters into the diploid stage, the 2N stage, and then occurs reduction division of meiosis so that the spores can be produced, which are haploid. Based on morphology of the mycelium, mode of spore formation, and fruiting bodies, there are 4 classes of kingdom fungi: the phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. Phycomycetes are also called as zygomycetes. They are found in aquatic habitats as they prefer moist and damp places. Some species are also obligate parasites on plants. The mycelium is aseptate and cenocytic. This means that the hyphae doesn't have cell wall and they are multinucleated. That means multiple nuclei can be observed in the hyphae. Asexual reproduction is through motile zoospores or through non-motile aplanospores. These asexual spores are produced endogenously in the sporangium. Sexual reproduction is through the fusion of 2 gametes, which could be isogamous or anisogamous or oogamous in morphology. Upon the fusion of gametes, a structure called zygospore is formed. Some of the common examples of zygomycetes are mucor, rhizopus, the bread mold shown here in the image, and albugo, that is the parasitic fungi on mustard. Ascomycetes are also commonly called as sac fungi. They are mostly multicellular, such as penicillium, and rarely unicellular, such as the most important and popular species, Yeast Saccharomyces. They can be saprophytic, decomposers, parasitic, or coprophilous. Coprophilous means that they grow on dung. Their mycelium is branched and septate. The mycelium will be having clear-cut cell wall and the uninucleate conditions can be found. There are also branches in the mycelium. Asexual spores for the reproduction of ascomycetes are known as conidiospores. Here in this image, we can see the conidiospores produced in the hyphae of Aspergillus. Sexual reproduction is through sexual spores called ascospores, which are produced in the sac-like structure called asci endogenously. These asci are arranged in different types of fruiting bodies called ascocarps. Some common examples are Aspergillus, shown here in the image, Claviceps, and Neurospora. Neurospora has been extensively used in genetic research. Many members, like morels and truffles, they are edible and they are considered delicacies. Here is the life cycle of Saccharomyces cerevisiae, the budding yeast, which starts with fusion of 2 haploid cells, and with their fusion, a diploid cell is produced, which later on undergoes meiosis to produce haploid spores again. They can also reproduce asexually through budding. Next category is basidiomycetes, that includes the most common and popular mushrooms. They can be found growing in soil, on logs, or on the tree stumps, and some members are even parasites such as rusts and smuts. The mycelium is branched and septate like the ascomycetes. Asexual reproduction is not found, but they can reproduce vegetatively by fragmentation. Sex organs are absent, but 2 vegetative cells or somatic cells of opposite strains and genotypes can fuse through plasmogamy to give rise to a dikaryotic stage that can ultimately produce basidium. Basidiospores are exogenously produced on basidium, arranged in fruiting bodies called basidiocarps. The most common members are mushrooms, scientifically called agaricus, astilago or smut, puffballs, and puccinia, which is also called as rust fungus. The last group is of imperfect fungi called deuteromycetes. Here, only the asexual and vegetative phase of reproduction is known. When the sexual form of these fungi are discovered, they are moved into the right class where they actually belong to. So, as long as their sexual reproduction, the way of sexual reproduction is not discovered, they are placed in deuteromycetes. It is also possible that the asexual or vegetative stage has been given one name and they are placed under deuteromycetes, and the sexual stage of some member is observed and they are placed into some another class. But later, when the links are established between the organism, the fungi is correctly identified and they are moved out of deuteromycetes. However, when they reproduce asexually, it is through conidia. The mycelium is septate and branched. Some members are saprophytes, some are also parasites. But a large number of imperfect fungi are actually decomposers of the litter, and they help in cycling of minerals. Some common examples are Alternaria, Calyptotrichum, and Trichoderma. This is all for the video. In next video, we'll talk about viruses. Stay tuned and please do subscribe our channel to support us. Thank you very much.